Got a biology final coming up? Then you are in exactly the right place. In this explainer, we are going to put together the ultimate study guide for one of the most classic topics in all of biology, bacterial gene regulation. Let's get you acing that exam.
All right, to really get a handle on gene regulation, you've got to start with the one thing that matters most to any living organism: energy. See, every single cell is working on a super strict energy budget, and pretty much every decision it makes is about being as efficient as possible.
I mean, just think about it for a second. Making proteins is a ton of work for a cell. It takes resources, it takes machinery, and it takes a whole lot of precious energy. So if a cell doesn't need a certain protein right now, making it would be like leaving every single light on in an empty house. It's just a complete waste.
And the short answer is, it doesn't. Yeah, no way. Cells are way too smart for that. The entire beautiful process of gene regulation is really just a sophisticated system designed to avoid wasting that energy. It makes sure proteins only get made when they are absolutely 100% needed. It's the cell's master plan for staying efficient.
So how does a cell actually pull this off? Well, you can imagine it like a complex genetic switchboard. It's not just a simple on-off switch. It's more like a whole control panel with all these different regulators that can fine-tune exactly how much of each gene gets expressed. Let's look at the two main types of controls on that board.
First up, you've got repressors. The easiest way to think about these is as the brakes. When a repressor protein latches onto a specific spot on the DNA, it literally gets in the way. It physically blocks the transcription machinery from doing its job. It's a stop signal, plain and simple. And then on the flip side, you have activators. If repressors are the brakes, activators are the gas pedal. When an activator protein binds to the DNA, it actually helps the transcription machinery get started, and it ramps up the whole process. It's not just a go signal, it's more like a floor it signal. So, what's telling these brakes and gas pedals when to work? Well, the answer is these little things called effector molecules. You can think of them like keys. They fit into the regulatory proteins, and by binding to them, they can either turn them on or turn them off, usually by changing their shape.
This whole key system leads to two major strategies. You have inducible systems, which are normally off by default. Think of these as the systems for breaking things down, catabolism. You only want to turn on the machinery to break down a sugar, for example, when that sugar's actually around. Then you have repressible systems. These are the opposite. They're normally on. This is perfect for building things, or anabolism. You keep the factory running, making something essential until you've got enough. Then you shut it down to save energy.
And this slide just shows it perfectly. Look over on the left at the inducible system. You've got a repressor blocking things, but then an inducer molecule, our key, comes along, pulls the repressor off, and turns the gene on. Now check out the right side. In the repressible system, the repressor can't bind on its own. It needs help from a co-repressor molecule to latch on and turn the gene off. Two really elegant strategies. Okay, all this theory is great, but let's see how it works in the real world. We're going to dive into the most famous example in all of biology, a true classic that you will definitely see on your exam, the lac operon in the bacterium E. coli. So an operon is basically a set of genes that are all controlled together as a single unit, which is just super efficient. Here are the key parts you need to know. The promoter, that's the starting line for transcription. The operator, which is the specific spot where the repressor, our brake pedal, binds. And of course, the actual genes themselves, which are called LAC-Z, LAC-Y, and LAC-A. And here's the really brilliant part. The whole operon gets transcribed into one single long piece of messenger RNA. It's called a polycistronic mRNA. What this does is it guarantees that the cell makes all three proteins needed for digesting lactose at the exact same time, from one blue part. It's like the perfect biological assembly line. Now for the genes themselves. This table is super high yield for your exam, so you'll want to know this. LAC-Z makes an enzyme that zaps lactose, breaking it in two. LAC-Y makes a protein that yanks lactose into the cell from the outside. And LAC-A, well, that one helps get rid of some toxic byproducts. And hey, here's a fantastic little mnemonic to lock this in for your test. Just remember what the operon does to lactose. It zaps it, yanks it into the cell, and acylates the leftovers. Z-Y-A. That's easy points right there. Okay, so the whole point of this operon is to deal with lactose, right? So it just makes sense that lactose itself is somehow the trigger. Let's see exactly how the presence of lactose manages to flip this switch on. When there's no lactose around for the cell to eat, it goes into full-on energy-saving mode. As you can see here, that LAC repressor protein is active, and it binds super tightly to the operator region of the DNA. It's a literal physical roadblock. RNA polymerase just can't get through. The system is off.
But when lactose enters the cell, everything changes. A tiny bit of it gets converted into a slightly different molecule called allolactose. Now this molecule is the true inducer. It's the key. It binds to the repressor, changes its shape, and makes the repressor just fall right off the DNA. The roadblock is gone. The path is clear, and transcription can finally begin. The factory is officially open for business.
Okay, now this is where it gets really interesting. Because it turns out lactose isn't the whole story. E. coli is a very efficient organism, and like a lot of us, it has a favorite food. And that preference adds a second, absolutely crucial layer of control here. This graph right here shows something called diuxic growth. See how the bacterial population grows, then it flattens out for a bit, and then it starts growing again? That's because E. coli will always use up the easiest sugar first, which is glucose. Only after all the glucose is completely gone will it bother to switch on the machinery to start using lactose. This is called catabolite repression, and it's all about using the best fuel source first.
So, how does the cell know that the glucose is gone? It uses a hunger signal called CAMP. The relationship is simple. When glucose levels are high, CAMP levels are low. But when glucose runs out, the cell gets hungry, and CAMP levels shoot way up. This CAMP then grabs onto our activator protein, the gas pedal, called CAP, and that whole complex binds to the DNA and supercharges the transcription of the lac operon. Okay, that was a lot to take in, I know. We've got the brake, which is the lac repressor, and we've got the gas pedal, the cap activator. So let's put it all together into one single cheat sheet that covers every possible scenario you might see on your test. This table is everything. It perfectly sums up the logic of the cell. But I want you to focus on that last row. That's the money shot right there. To get really high, roaring levels of transcription, you have to meet two conditions at the same time. The brake has to be off, meaning lactose is present, and the gas pedal has to be floored, meaning glucose is absent. Any other combination, and you get little to no transcription. This is the absolute key takeaway for your exam. All right, before we wrap up, let's go over 3 super common mistakes students make on this topic. You're not going to make these mistakes. First, remember it is allolactose, the isomer. That's the real inducer, not lactose itself. Second, don't mix up the two controls. The repressor responds to lactose, the activator responds to glucose. And third, remember that the operon is never truly 100% off. There's always a tiny bit of leaky expression, which is how the cell can get that first bit of lactose inside to start the whole process.
The lac operon is it's a masterpiece of cellular logic. It shows how elegantly a simple cell can respond to its environment to save energy and maximize its chance of survival. And it really leaves you with a final thought. If this is what one bacterium figured out, what other incredible, smart systems are still out there just waiting for us to discover?